Okay, so for lab practical 2, we're going to go over first part today. Start with moving across a membrane, specifically diffusion and osmosis. So diffusion is just movement of material from an area of high concentration to an area of low concentration. So in this case, you take a sugar cube with coloring in it, put it in the sugar cube. You start out, you have a high concentration of sugar in one area, low concentration in another area. Over time, the concentration gradient gets eliminated. You get equal distribution. Okay, that's diffusion. One thing I want to note to you is that temperature can increase the rate of diffusion. The warmer the environment, faster diffusion is going to happen. Okay? Basic principle. So another principle of diffusion is that diffusion increases with decreasing molecular size. So here's 3 solutions that are in a petri dish that can diffuse through the petri dish. It's diffusion of solids. So potassium permanganate has a molecular weight of 158. Ferric chloride has a molecular weight of 162. Methylene blue has a molecular weight of 319. Okay, if you look at diffusion over 60 minutes, okay, this is the one that diffused the most, potassium permanganate. Something they also mentioned here is that it's also the one with the smallest molecular weight. Okay? So smaller objects can diffuse faster than bigger ones, right? Because it takes more to move them. Okay? Now, diffusion of water is osmosis. So one of the things we want to point out to you is you have different kinds of solutions: hypertonic, isotonic, and hypertonic. So in a hypotonic solution, hypo means lower, hypo for low. It means the concentration of solute is lower outside the cell. It's lower outside the cell. So that means the concentration of solute is higher inside. It would want to go from concentration of high to low, which would make it go outside, but the membrane is impermeable to that solute. So water is going to go the opposite direction. So the solute would want to go out, water is going to come in. Okay, it goes the opposite direction. The other way of looking at it is, if the concentration of solute is low outside the cell, concentration of water is high. And the water is going to go down its concentration gradient and go in the cell. In the isotonic solution, iso means same. So there's no net movement. There's still movement, it's just no net movement. So if we give you on a test, practical, here's the solution, draw the arrows or draw what's happening. For the hypotonic solution, you draw water going in. For the isotonic solution, you just draw equal signs to indicate there's no net movement, it's equal movement in both directions. Now, for the hypertonic solution, hyper means higher, right? Like hyperactive, really active. So you're going to have a higher concentration of solute particles outside the cell. They would want to go in. They can't, so the water is going to come out. So in that situation, you can also look at it as the concentration of solute is higher outside the cell, the concentration of water is low. Water is going to want to go toward the low concentration, so it goes out. So either way, whichever way works for you, but in this case, you draw water coming out. Okay? So just practice that, okay? think we have some games, purpose games that you can do to practice that as well. All right, here's a clinical application. Again, no net movement, equal movement in both directions in the isotonic solution in a red blood cell. Normal shape. For a hypotonic solution, again, the concentration of solutes higher inside than outside. So it's a hypotonic solution. Concentration of solute outside the cell is low. Hypo for low. Means concentration of water is high outside. So it's going to go in. Cell takes in the water, expands, and can eventually burst. So, in the opposite, or it swells, is one way to look at it. In a hypertonic solution, concentration of solutes high outside the cell, concentration of water is low. The water is going to go out toward the low. So, here, what we call that is shriveling or crenating. Okay, so crenation is what happens in a hypertonic solution. Okay. So that's clinical application. Okay, let's get into histology or study of tissues. So let's start. You have four categories. Remember your big categories: epithelial tissue, connective tissue, muscle tissue, nervous tissue. Our plan today is to cover epithelial and connective tissue. So for epithelial tissues, you can classify them by a number of layers and the shape of the cells. If there's one layer, it's simple epithelium. If there are multiple layers, it's stratified. One layer simple, multiple layers stratified. Now for shape, you look at the shape of the cell facing the apical surface. So if the shape is flat, it's squamous. Squamous can also look like a fried egg. Okay, so squamous could also look irregular, like that, depending on which angle you're looking at it from. So if you're looking at it from the side, it'll look flat. If you look at it from the top, it'll look like a fried egg. If the shape is more cube-like, it's cuboidal. If the shape is more rectangular, it's columnar. Now for stratified epithelium, you look at the layer at the apical surface. So here's your apical surface. It's not touching a basement membrane. These are flat, so it's stratified squamous. Doesn't matter what these are shaped like. It's not the apical surface. Here, cells on the apical surface are cuboidal, so it's cuboidal, multiple layers, stratified cuboidal. Here's the cells on the apical surface are rectangular, multiple layers. It doesn't matter what shape these are underneath. It's stratified columnar. Let's see. All right. Well, not doing that. All right. For simple squamous, here's a view of some simple squamous cells. Got some of these respiratory tract, the alveolar air sac walls. Very irregular shapes, and one layer. And these are just one layer of cells. help with absorption, filtration, diffusion. Right? You want diffusion at your air sacs. You want to be able to exchange oxygen and carbon dioxide, right? Here's another view. Bowman's capsule on your kidneys. We're not looking at everything. We're just looking at these cells surrounding the nephron. So it's Bowman's capsule right there. These are single layer of flat cells. Okay? So that's simple squamous. All right, here's another view, simple squamous epithelium. This one is looking at it from the fried egg view, looking at it from above, essentially. This is also simple squamous. So just either way, just know you could see it in either way. You could see it at one slide, it could look like this, one slide could look like that. Now for stratified squamous, this one's in the esophagus in your digestive tract. Again, multiple layers, different shape down here, but the only thing that matters is that these are flat at the apical surface. And you see these in the esophagus, the mouth, vagina, epidermis. All right, stratified squamous, another view of it, okay? Multiple layers, flat cells at the apical surface. Multiple layers, flat cells at the apical surface. All right, simple cuboidal. One layer cube-like cells. Help with secretion and absorption. See these in the kidney tubules. See these in some glands and in the surface of the ovaries. So you can see here, you have one layer cuboidal shaped cells. Here's another view. You have these circles, but it's just one cell layer thick and they're cube shaped. You see that here as well, one layer, cube shaped shells, or cube shaped cells. All right. Stratified cuboidal. These are protective cells. You see these in sweat glands, mammary glands, salivary glands. Multiple layers cuboidal cells. So here you can see at least one layer here and then another layer around it. So stratified. Okay, another view. More than one layer, cube-shaped cells. More than one layer. So here's sort of an inner layer and an outer layer. Okay. Stratified cuboidal. All right, multiple layers. Oh, this one is simple, so it's one single layer. One single layer, rectangular cells. This can help with absorption, secretion. One thing I want you to know here is these circles that are light stained, these are goblet cells. So G-O-B-L-E-T, goblet cells. And they secrete mucus, these goblet cells. See them in the digestive tract, gallbladder, bronchi, uterus. Okay, one layer. Rectangular shaped cells, simple columnar. Again, one layer, rectangular, simple columnar. And you can also spot the goblet cells. So that's a structure that you might have to spot on a practical. So you might have to say what those goblet cells produce. Okay. Okay, for stratified columnar, you have multiple layers. Here's one layer, there's a layer underneath it. With the layers of the apical surface, The ones that are exposed, the layer that's exposed, cells are columnar. They're also involved in protection and secretion, but you don't see these very often. Involved in male urethra in the reproductive system, as well as ducts and glands. Okay, stratified columnar epithelium. Another view, multiple layers, but the cells at the apical surface that are exposed are rectangular. All right, so if you ever see on a slide, or it looks like some cells go all the way to the apical surface, some don't, that's pseudostratified columnar epithelium. And in this case, because they have cilia, these little hairs, it's pseudostratified ciliated columnar epithelium. Okay, but all the cells touch the basement membrane, but some of them don't reach the apical surface. All right, and these are usually involved in some sort of secretion. See these in male reproductive system, some ducts, respiratory tract, trachea. All right. So if you see the cilia, it's a clue. It's probably the pseudostratified columnar epithelium. Not always, but in many cases it will be. And then here again, you can see another goblet cell. Okay. All right, transitional. Transitional is going to have different shapes. Like here, it's going to be all pillowy because your bladder is not full. Okay, it's floating. All right. So sometimes it can resemble stratified squamous or stratified cuboidal, and other times it's going to be more flat, depending on if the bladder's full. All right. More views of transitional epithelium. But mainly know it's associated with the urinary bladder. Okay. All right. So it looks like every table just about has at least one person. So I'd like for you to talk with your partner real quick and just talk to them about epithelial tissue, simple versus stratified, squamous versus cuboidal, voice versus columnar, and some of the different types of epithelium you saw. All right, so let's cover connective tissues. So one type of connective tissue you'll see is mesenchyme, or mesenchymal cells. These are embryonic connective tissue, so these are in embryos. All right, so you'll see in these tissues, these are ones that give rise to the other connective tissue types. You'll see mesenchymal cells, you'll see ground substance, you'll see some fibers. To be honest with you, you'll probably tell this one, if you see this on a practical, you'd probably tell it by making sure it's not anything else, okay? The others sort of have these hallmark things that you know, okay, I see that, I know it's this. You know, and so mesenchyme tends to be one where you don't see that because it's developmental, right? You want it to be able to differentiate into different kinds of tissues. Yes?
So are you saying that these ones are all stem cells?
Yes. So you wouldn't, because they're stem cells, you're not going to be differentiated into a bone cell or a dense regular connective tissue cell. So you want something that's very generic, where you can turn it into what you need to. Okay. All right, here's some more views of the mesenchymal cells. And this one's from an embryo, and this one's from an umbilical cord. Okay, same pictures. All right. So, like, notice how I said that one's very general. The others usually have some sort of hallmark feature that you can really tell. So, next connective tissue we're going to cover is reticulum. And let's see if I can find room. So, one of the things I also want to make sure you're aware of is the big categories for connective tissue. Let's see, I have them written on the side. No, I don't. So, for connective tissue, we have connective tissue proper. Fluid connective tissue, supporting. All right, now connective tissue proper is going to have some subcategories. We'll get into those in a minute, but those are sort of your big well, technically, connective tissue is your big category. These are subcategories. So when I said earlier, you would have to know specific tissue, big category, subcategory. Put it over here. CT proper, connective tissue proper, fluid, supporting. So, connective tissue proper has some more headings underneath it, but we're not going to worry about that too much right now. All right, so in connective tissue proper, what I will tell you is there's loose connective tissue and dense connective tissue. Reticular connective tissue would fall under loose. How you know, if you look at this slide, if you know that this is reticular connective tissue, these black fibers. They're called reticular fibers, reticular tissue. So if you see the black reticular fibers, it's reticular tissue. So another loose connective tissue is adipose tissue. Adipose is a fancy anatomy word for fat. These are fat cells. You'll see these under the skin, around kidneys and eyeballs, your abdomen, okay, breast area. So the adipose cells are like big circles. The nuclei, you know, in most cells, the nuclei is sort of somewhere in the middle of the cell, right? In a adipose cell, the nuclei are along the edge. So when you see these little dark spots on them, those are nuclei. Okay, the rest of the cell pretty much is taken up by the fat globules. And what are they for? Well, they store fat. But if you see the big circles, adipose tissue. Okay. Black lines, reticular fibers, big circles, adipose tissue. Here's some more views of adipose tissue. So the lining of the circles, that's your cell membrane. Fat droplets in the middle, nucleus on the sides. Another type of loose connective tissue is areolar tissue. So an areolar tissue you're really going to see this, and it's going to have this kind of combination of different kinds of fibers. You'll see the big pink collagen fibers, the little elastic fibers. The little dots are your nuclei of your fibroblast cells. Remember, these make your collagen. All right, in this picture, what I really want you to see is this layer is your epithelium, and underneath it, you have a layer that we call the lamina propria. So you'll definitely want to know that term, or lamina propria, which is connecting the epithelium to the tissue underneath. All right, so that's loose connective tissue. Let's get into dense connective tissue. So in dense connective tissue, you can have dense regular connective tissue. Looks very wavy. Okay, very regular pattern. All right, dense regular. All the pink is collagen fibers. The little dark lines, those are your fibroblast nuclei. Where do you find dense regular connective tissue? Tendons, ligaments, the flat sheets called the aponeuroses. And they are attachment sites. And you should be able to tell this is much less of a regular pattern, correct? Doesn't look as wavy, right? Yes or no? Can you all look at the picture? Does this look as wavy as the previous one? It's less regular, so it's dense irregular. Still dense, it's still packed together, but it's irregular shape. Has some elastic fibers. See it in the fibrous capsules of organs and joints, in the dermis, the submucosa of the digestive tract. All right, another dense connective tissue is elastic tissue. And the difference between this and dense regular is the presence of all of these. It does have more of a regular pattern, but it has all these dark lines in it. And those dark lines are elastic fibers. So these are not reticular fibers. The reticular fibers are kind of like a network. These are more like lines, right? They just go across. Those are elastic fibers. And elastic fibers are stretchy, they recoil. Where are you going to have those? Places where you need stretchiness. Like your aorta, your heart, in the walls of your large arteries, bronchial tubes. Okay, another view of your elastic tissue. So here you have the wavy lines. So this is at a higher magnification. So if you see the wavy black lines, it's elastic tissue. And the space in between the elastic fibers is ground substance. All right. So now we're moving in away from connective tissue proper to the supportive connective tissue, to support it. One of the examples of supporting connective tissue is cartilage. One of the types of cartilage is hyaline cartilage. So this is a specific type. And hyaline cartilage I would say of pretty much all the cartilages, you'll see these chondrocytes in their lacunae. So if you see these, to me, they look like eyes. Okay, if you see these things in there, that's cartilage. These are chondrocytes. So if you see chondrocytes, cartilage. Space around the chondrocytes is matrix. Where do you find hyaline cartilage? Embryonic skeleton as you're developing, as an embryo, long bones on the ends of your long bones, your costal cartilage in your rib cage, cartilage in your nose, in your trachea, and your larynx. All right. Usually, depending on what you stain it with, it has this pink stain. All right, now here's another example. Again, you can't memorize it pink because if you use a different staining agent, here it's staining more blue. But again, you see all these collagen fibers, okay? So this one is different from hyaline cartilage because here you don't see any fibers, right? You just see matrix. Everybody look at the board. So the board of the screens. Hyaline cartilage, you just basically see chondrocytes and matrix, right? Now, if you introduce fibers, okay, it's a different kind of cartilage. In this case, it's fibrocartilage because a lot of the collagen fibers, right? Intervertebral disc, pubic symphysis, where you connect the pubic bones together, discs of your knee joint. Now, for elastic cartilage, looks a little different from fibrocartilage and from hyaline cartilage because you've got these dark elastic fibers in it. It's still cartilage. Why? Because you see the eyes, right? The chondrocytes in their lacunae. It's elastic cartilage. Where do you find elastic cartilage? In your ear, external ear, right? It's very stretchy. It recoils. The other kind of supportive connective tissue is the bone. So if we do what kind of plane is this? Remember? Transverse, right? Cut through. Look at the inside of the bone, cross section. You'll see in the outer layer, spongy bone. The outer layer is compact bone, the inner layer is spongy. Okay, inner layer is spongy. All right, outside of your bone's hard. It's compact, right? Yes, no? Yes, outside of your bone's hard. Inside is spongy. So this little network looks like spongy, so spongy bones. Outer layer of your bone forms an outer layer called periosteum. The periosteum has a fibrous layer on the outside, cellular layer on the inside. One of the big characteristic features, you know you're looking at bone, are these rings. You see the rings, you're looking at bone. And the rings, if you see basically a unit of rings together, a circle with a bunch of circles in it, that big circle and everything in it's called osteon. Okay, that's an osteon. So in the center of the osteon, we have what we call a central canal, and there are blood vessels in there to give the bone its blood supply. Now, these little dark spots are kind of in a circle pattern around the central canal. Those are osteocytes, and they also live in lacunae. Okay, so the dark spots are the osteocytes. And you have these little faint lines coming off of the osteocytes. Those are canaliculi. Now, the general space knot stains, matrix. All right. You also have these rings. If you look at the rings, in this view, each circle is called a lamella, plural lamellae. They're concentric, all right? Form circles over each other, concentric lamellae, okay, till you get to the central canal. Here's another view from the side of the compact bone, another view of the spongy bone. Okay, but these are all fair game structures we could ask you. Okay. All right, so that was supporting connective tissue. Here's fluid connective tissue. First type of fluid connective tissue is blood. Lots of different blood cells, right? Most of these little red circles with the light spaces in the middle, so red blood cells or erythrocytes. Carry your oxygen. This little dot here is a platelet. Okay, the other cells are white blood cells, different kinds of white blood cells. You have neutrophils, okay, have sort of like a, I don't know what you want to call that, like a backward C or whatever in there. It's a neutrophil. Or just, basically you've got different kinds of block shapes inside. So if you have a big circle, it's a monocyte, all right? If the circle's taking up most of the room in the cell, it's a lymphocyte. Discrete dots that are really, really dark, the basophil. If the dots are really small, it's the eosinophil. Now, what I really need you to know about this is not necessarily each of the different types. I mean, I can ask you that, but really what I want you to be able to tell me is this a red blood cell, a white blood cell, or a platelet? Okay? So, based on what I've told you, do you think you can tell the difference between a red blood cell, a white blood cell, and a platelet? Yes or no? Hands up if you think you can tell me the difference. Okay. I don't see a lot of hands up, so that tells me we need to spend a little more time on it. We got red blood cells. They're kind of purplish, white blood cells. It's a little speck of purple, platelet. All right, what's this? What's this? Platelet, right? Okay. You good? Okay. All right, so that's all right. So I think I will skip ahead just a bit past the tissues. I think yeah. So we'll pause there on the video. So what we're going to do in class is we're going to look over our slides that we have at our tables. Okay, we're going to get them focused, and then we can go around and you can take pictures of every slide. Okay, and we'll match the slide with the white piece of paper. Okay. If you're watching this at home, what I'd like for you to do is to go to Practice Anatomy Lab. It's in Mastering. You look for, once you go into Mastering, look for Practice Anatomy Lab and click Histology and look at tissues. And so you'll go through and you'll practice. You can do a practice quiz or flashcards and go through and study the different types of slides. You can also go to Purpose Games and do the histology games that are in the modules. All right. Let's see.